Good morning, everyone. We will let people trickle in here. Shortly, we'll begin. Just to give it a few minutes to get everyone into the window. Thank you so much for joining. Let's go ahead and kick it off. I see that people continue to trickle in, but they'll kind of trickle in as we intro here. So, hi, everyone. Thank you so much for joining. My name is Tyler Casey, Senior Marketing Specialist here at Bellsera. And thank you for joining, as a part of our ongoing monthly webinar series. Today, I am joined by our Chief Science Officer, Jack DiGiovanna, to talk and learn about enhanced genomic accuracy, superior alignment, and efficiency gains. So Jack will dive into those topics as we move into the presentation. It's my pleasure once again to introduce Jack. Jack plays a pivotal role in shaping the scientific strategy across various sectors, including government, biotech, and diagnostics. With a strong foundation in academic research and extensive experience in developing data ecosystems, Jack excels at translating complex scientific concepts into actionable insights for diverse audiences. His leadership has been driven groundbreaking advancements in biomedical data ecosystems, and he is recognized contributor to numerous NIH projects from Kids First to the Cancer Research Data Commons. A passionate advocate for translational research, Jack is dedicated to empowering researchers and accelerating scientific discovery to improve patient outcomes. Just want to shoot one friendly reminder throughout the presentation. If you have any questions, feel free to drop those into the Q&A of the Zoom, and we'll chat with those following the presentation. Jack, I'm going to pass it over to you.
Thanks so much, Tyler, for that very kind intro. Just want to check that you can hear me okay?
Yes, perfect.
Perfect. Thanks, Tyler, and thanks everybody for coming and spending some of your time to learn a bit more about graph genomes. And you may be wondering, why is there a picture of a rear cassette on a bicycle on the title slide? What does that have to do with graph or anything else? Is it just because Jack loves cycling and is always on his LinkedIn? Well, that is part of it. I do love cycling, but actually, coming back to my time as a postdoc and group leader, one of the talks that really resonated with me, they were looking at biomechanics and different efficiency of vehicles. And what they found is that the most efficient mode of transportation for humans is a bicycle. You can get around estimated 3,000 miles on the equivalent of 1 gallon of gasoline. So I like bikes, but also it has a nice tie to efficiency. And efficiency is one thing that we can certainly talk about when it comes to graph genome. But I'm actually going to pivot a little bit and start more with the impact. My clicker would work. Sorry. And start with the impact. And really frame a lot of this talk around tumor mutational burden, because accurately estimating that can extend patient lives. And this is especially impactful for underrepresented populations. So what I mean? There's a great paper that came out about a year and a half ago, driven by a group of authors from Dana-Farber. This is the very first sentence of that paper. So this immune checkpoint inhibitor, this is actually Keytruda, Merck's Keytruda, which is a blockbuster immuno-oncology drug. I think it's approved now for 17 or 18 different cancers. Bristol Myers Squibb's Opdivo would be a similar drug with this mechanism of action. The FDA approved for solid tumors with a high TMB. So that conditional is really important. Why? Well, because TMB is often overestimated in non-European populations. So if you look at these three patients here, let's focus on the top one first. So this first patient, self-reported white, has 2 true somatic variants and 2 that are germline filtered out. So this person's true TMB is 2 and the estimate is exactly 2. All good, exactly how we would design this. Contrast that with this self-reported black patient who has 1 true somatic variant, but 3 germline false positives get through. That means this patient's estimated TMB is 4 times higher than it actually is. What are the impacts of something like that? Well, they can be quite stunning. So the authors of this paper looked over 1,000 patients who were treated with immune checkpoint inhibitors. And here's a survival curve. And as you would expect, based on the indication, the red group that has higher tumor mutational burden has a survival advantage over the blue group with low TMB. But what was quite problematic is this orange group, which is disproportionately Asian or African American, their estimated TMB was high, but their actual TMB was low. And since their actual TMB was low, they fully overlap with the blue group, whose TMB is low. There's no survival advantage that they're conferred. And think about the impact here on the patients. They were told that they were a good candidate for this immune checkpoint inhibitor as a way to extend their lives, as a way to improve their lives. And actually, they had very poor outcomes. Zoom out also for someone who's developing a drug. They were trying to design a therapy and specifically test its impact and effectiveness on folks with high TMB versus low TMB. And they may have included a whole group of persons into their trial, which added a lot of noise, lowered signal to noise ratio, created a longer, more expensive trial with less clear results. So with that intro, for me, the reason that I'm come to work every day and I'm excited about what I do, and I suspect a lot of you in the audience as well, and a lot of my colleagues here at Belsera, is that precision medicine can create amazing outcomes. Some variants of cancer are effectively cured, but that's only for some patients. Right? And one of the reasons behind that that's been well understood over the last few years is that we need as a field to better understand diversity and better represent diversity in our analysis. That comes to the fact that precision medicine over the last 20 years has mostly been driven by a linear reference created by a single, 70% of that coming from a single white male of European descent in Buffalo, New York, somewhere here in T-SNE space. Now, for me, as a white male of European ancestry, probably fine. I'd be okay. But clearly, if I were Finnish, Korean, African or African American, I'm now getting farther apart in T-SNE space, and we could debate over a beer what that means, but clearly there's a clustering of haplotypes and genetic information that are not very well represented using a reference from here. And that comes back to those false positives that we saw in the third patient a few slides ago. So our company has been passionate about this for more than a decade, right? One of the companies that came together to form Belsera was Seven Bridges Genomics. The name of that company is based on the Seven Bridges of Königsberg math problem proposed by Euler that showed the shortest way to cross all bridges is not something that you can solve with standard mathematics. You needed to use graph theory. So we've been pioneering graph-based pangenome-based references since 2014, have finally got it onto the market this year as a robust, scalable, and validated pipeline that's ready for folks to try. And it's not just our excitement or our internal bias that's saying these things are important. About 3 years back, the Human Pangenome Reference Consortium was formed, and they published their first pangenome reference in the middle of 2023. So it's becoming state of the art and more recognized as a standard way to do alignment. So for those of you who aren't familiar how graph works, I have a few simple illustrations here that hopefully will make it clear. In this case, along the top, we have reads that are aligned with a graph reference. On the bottom, the same reads that are aligned with BWA and a linear reference. What you can see here is that both of them find an insertion, but graph correctly recognizes that it's a homozygous insertion where the linear reference estimates that it's heterozygous, and also look at the overall alignment and clarity around the insertion that you have on the graph reference. It's very high read depth. You don't really have that. You have quite a bit of noise all around it in the linear reference. That noise around it means that graph is going to excel also at identifying complex variants. So when you have that SNP close to an insertion or deletion, graph has no problem with that because they're represented independently, and there's a very clean alignment to the insertion, where a linear method could struggle with that because they would have a lot of noise around these regions and low read depth. And finally, compound variants follow the same exact logic. In this case, when you have a SNP inside of an insertion, you would be able to represent both of those things on the graph and able to map that quite clearly. What's really neat, and we're going to come back to this later, is that this is all represented in one structure. It's all calculated in one pass. And the actual shape and width of the graph actually represents the population diversity and population characteristics. So we'll come back to that a little bit later. So these are all beautiful pictures, really nice to show, I hope hypothetically, why you get, hey, PowerPoint science-wise, this should work. We should detect more. We should have better read depth. We should have higher accuracy. But the natural question would come up is, well, who cares? Does it matter? How do you test that this has an impact? So we took that challenge in bringing this to BioIT earlier this year and defined 3 metrics that we believe would be important and based on the interactions we've had with the community would be important in showing that graph actually is pangenome references can be a game changer. These 3 metrics are accuracy, cost effectiveness, and compatibility. I'm going to show them in the next few slides. So starting with best-in-class accuracy. Graph performs exceptionally well at detecting structural variants. So here on the top right, we have a precision along the x-axis, recall along the y-axis plot. I included the equations for precision and recall just in case anyone's not familiar, and because we're going to show a couple of these plots over the next few slides. But here, graph in green is compared to GATK, deep variant, dragon, so compared to multiple best-in-class solutions or leading solutions. And what you can see along precision, these are all relatively similar, with graph performing slightly worse than precision. But in recall, having over 3 times better recall than the other methods. And what's also exciting is looking at the characteristics of those SVs that are called. So here on the bottom right, on the y-axis is, whoops, sorry about that, new computer, so it advanced the screen for me. On the y-axis is the number of SVs that are detected, and on the x-axis is the length of those structural variants. What you can see compared to these other leading methods, graph is identifying 10 to 20x longer insertions or deletions than the other methods. And look, staying with SVs for a minute, the performance improves by using representative genomes, as you would expect, representative references. So here I'm showing 3 different plots. One is an African sample, one is a Brazilian sample, and one is Native American. Along the y-axis is the SV count. And in this case, in gray, we have the state-of-the-art linear aligner, GATK with BWA, GRCH 38. Comparing that to a pangenome global reference, so including all the populations, and we'll come back to the composition of these references at the end of the presentation, but including all the references, what you can see, at least in these 3 samples, is you get about a 2x improvement in the number of SVs that are detected. Now, if you go a step farther and use the relevant population reference, really focus on that using the African reference for the African sample, you see an additional 2x improvement in the number of SVs that are detected. And there's no shenanigans going on here where we actually constructed the reference using the sample that we're aligning to. It's just having more representative shape and dynamics of the reference compared to that's more closely associated with the samples that are being aligned. So this works really strongly in detecting more SVs. The next question, of course, is, well, if you can detect more SVs, that's great, but where are they? Do they matter? Would this perform well in challenging medically relevant genes? So here we're getting into some genome in a bottle samples. On the top, we're looking at indels with precision and recall plot, and on the bottom, we're looking at SNPs. And what you can see, graph again in green with the competitors in the other colors, pretty similar in terms of precision, but graph able to call about 5% more indels than other solutions. That means that the false positive rate is quite low, which is going to optimize variant yield and reducing downstream work to correct that. Finally, what about a really difficult region? So the major histocompatibility complex, really important for immune system function, also hugely polymorphic and quite difficult to map. There's complex SDs. We participated in the Precision FDA Truth Challenge, the one that ran recently. And we're able to show in this particular region, both for indels and SNPs, we had the best performance across all of competitors, which would mean we are the farthest to the top and right in all plots for this region. So it performs better with SVs. It performs in regions where it matters. It performs well in very difficult to map regions. Let's come back to the original question and that survival curve of what about this issue where if you just estimate TMB with tumor, you're going to overestimate the TMB score, especially for underrepresented populations. So we have 8 samples here. We'd like to expand this in the future, but we took 8. And here we're looking at TMB scores on the y-axis. And you could see here, if you process with GATK just with the tumor sample, you would get a score of 40 for this individual. And if you had tumor and normal, and having tumor and normal is going to allow you to filter out some of those germline false positives, it adjusts the score down to it's actually around 25. And you can see it's different across different samples. In some cases, it's quite close, actually. In some cases, it's dramatically overestimated. There's over on the right side, we have the African samples. They seem to be more overestimated than the European, but small sample size and all of the usual caveats. But just if we work with what we have, what does it look like if we use graph with the tumor-only samples? What that looks like following the same conventions is with tumor-only and graph, the TMB score is much closer, at least in this sample, to the tumor normal corrected with linear than linear tumor only. And what we did was to normalize this so it's a little bit easier to see. In this case, all results have been normalized to the tumor normal TMB score with GATK. And you could see there's clearly different distributions between the tumor-only process with graph and the tumor-only process with GATK. And further, as expected, the value of tumor-only process and graph is much closer to the GATK tumor normal. So hopefully that gave everybody a taste of the accuracy. But if it costs $1 billion, what does it matter, right? So the next thing that we needed to show was cost effectiveness. Getting right to the point, these pangenome methods that we've created are faster and cheaper than the field. So on the left, we have duration. On the right is cost. If you compare any of the green methods, the pangenome methods, to GATK in orange, it's about half of the time and about 2/3 of the cost. Now, if you look within the pangenome methods, there's 2 here at the top. So graph is the method that we have developed over the last 10 years at Valsera. Pangenotyper, we co-developed with Sention, which is using similar technology. Both of those are both faster and less compute costs than VG and deep variant or VG. These methods are all optimized in-house by our expert bioinformaticians to the same level of quality. So this is not that we had like a grad student come in and try to optimize VG just so it would run faster. This is considerably faster than we've seen in the field or from competitors. So we've really got this down as much as we can, but it's still much more efficient or more efficient to use graph or Pangenotyper. And the next thing that comes up is pretty neat. As I said in the beginning in those graphical overviews, the shape of the graph is actually capturing the posterior probability. What does that mean? Well, it means that just by running the graph-based aligner invariant caller, you can get a lot of the sensitivity of joint calling and natively call about 80% of the variants that would be rescued by GATK joint genotyper without having to do joint calling. And similarly, you can filter out about 80% of the variants that would be filtered out by the QSR. They're just not called by the graph alignment and variant caller. So that seems good, but let's make that a bit more concrete. We worked some years ago with partners who were working on cardiac defects in pediatric patients. They were working with the bench to bassinet data set, which was co-collected across NHLBI and Common Fund, which means half of the data set was stored in one place, half was stored in the other place. Those places were analyzed, the data sets, but what we actually needed to do was to bring them all together and do joint calling to actually capture the whole population. So that was possible. That was done. I think, on the Chromatica platform by our collaborators at CHOP, and it was fantastic and really excited. It led to multiple publications. But bringing that data together required quite a bit of interoperability to be created. And I think just the compute cost, nothing else, just the run to do the joint calling was between $40,000 and $60,000 of compute. So pretty big deal. And now think about it as we get to more situations with enclaves, as you get to situations where data is has particular privacy restrictions and can't move from the location that it is. Well, that becomes real problematic for joint culling because you can't actually combine the data together and bring it to one location. It has to stay where it lives. Graph can be a really powerful way to capture much of the posterior probability just within the reference. So you actually don't need to do the joint calling. Much more work can be done to establish that, but hopefully that gives a flavor of the real possibility there. The other thing where graph is quite powerful is trio analysis. So here we compared a number of different workflows, best in class, and looked at the graph trio and Pangenome at the top compared to giraffe, BWA, BWA-GATK, Kids First Consensus. And what we found is comparable results to state-of-the-art workflows, but as little as 1/3 of the cost in time or money. So it can really move the needle forward there. So hopefully that's shown from a lot of different facets that this is a pretty cost-effective approach. It's faster, uses less resources. But we knew from the beginning, this has to be compatible. We can't define some exotic standard or require you to rip and replace your entire bioinformatics pipeline, right? People are trying to get best-in-class tools and combine them for their particular research questions. So from first principles, we said this has to be fully compatible with the rest of people's research flows. And that's what we have, just to make this a very short story, right? So you will take in FASTQs, CRAMs, and BAMs as your input to the aligner, just as you would for a linear aligner. After alignment, you get out BAMs and CRAMs. If you use the graph-based variant color, you'll get out VCFs. That means you can pick and choose which pieces you want to use. You can then, from the VCFs, add your own annotation, do your own tertiary analysis, whatever your secret sauce is that's important to drive forward your research, fully supported and compatible without you needing to write a bunch of converters or maintain 2 versions of things or other fun and games like that. Further, you get a nice user-friendly experience. This is an example of our graphical workflow configuration when you're setting up a task. You have your inputs on the left, app settings in the middle, and I think outputs are going to be on the right. You can see the sort of node and a wire diagram way to represent the inputs and outputs. So if you're running this on a Bellsera platform, you would be able to have this experience. Now, I know some of you in the audience might be looking at this and absolutely cringing and all the clicks and visual, and I don't want to do this, and I have to run this on 10,000 samples. Great. You can also do this completely by API, set up a batch job, and just have it run and not look at anything graphical at all, except when you get to the results. And when you get to the results, as I said, they're fully compatible. Here's an example of taking those reads and putting them in a Jupyter notebook. You could also have this in RStudio. You could have it in some of our platforms. You could have this in SAS. You could also take the BAM file and put this out to IGV to be able to see, the full reads and where the variants are. And again, continue along your merry way with whatever other bioinformatics you want to do downstream of this. So what's in the box today? What do we have available? If you act now, what can I get you lined up with? Well, showed in this presentation already, the African, Brazilian, Native American, and global references. In total, we have 9 population-specific references and 1 global reference. The global reference represents about 800,000 total samples. including 47 whole genomes from the Human Pangenome Reference Consortium. And if you want to know how these are built, here's an example for the African Pangenome Reference. Starts with about 21,000 SNPs and indels coming in from Nomad, and then iteratively add about 500 African participants from 1,000 genome to build out the SNPs and indels within the reference. SVs, similarly, we start with 10 human genome structural variant consortium references, and then iteratively add in about 400 African participants from 1,000 genomes. So that's how we can build this and capture a lot of that diversity, building on the great work that the Human Genome Structural Variant Consortium reference is doing, and also bringing in data from other sources as it's available. So what's the call to action? Well, I firmly believe that graph can improve your business outcomes. I think it can drive forward precision medicine. I think it can better represent diversity. But it's not important what I believe. It's important that we can show this actually matters for your application. One of the challenges is not having to work with genome in a bottle data sets. Right there, you have ground truths, but you'd actually like to say, what does this matter? How does this impact things in the field? So what the ask would be is that you reach out to us. I'm sure Tyler has great links in the registration, and they'll be reaching out to you to say hi afterwards, but that you're able to work together for about a 4-week pilot where we would detail with your team what are success criteria that would actually move the needle for your business. We've worked with pharma, with biotech, with academics, right? Don't feel boxed in by, okay, we're only in this group and this is only for drug discovery, right? So I think that's one of the main use cases, but if you're interested, please reach out to us. Then we could test your data, or sorry, you could test your data, excuse me, either on one of our platforms, which I showed a few slides ago, or on-prem. And then after the end of 4 weeks, we'll get together. Did the thing move the needle like we thought it would? If it would, let's move forward and help advance precision medicine. So why I really hope you use a POC using pangenome references with tools like Pangenotyper or Graph, it's going to get you to best-in-class results. You'll have less false positives. You'll have higher signal to noise. You'll just have better alignment and variant calling. And it does this efficiently. It's not going to take more time. It's not going to take more resources. And it's fully compatible with your existing flow so that you can drop it in, just drop in the pieces that you need to move things forward for your business. But most importantly for me is you'll better represent population diversity. You'll have less false positives. You'll find previously unseen variants. And think about the impact that can have for a patient, the analysis that they get, the panel that they run so that they can understand, yes, I'm actually a great candidate for this therapy or not. And also taking, turning around and taking the perspective again of drug development, when you can actually be more confident in the biomarkers that you're using and in the different values, thresholds that you're using to define inclusion and exclusion criteria so that you will design smaller, less noisy, shorter clinical trials. So I've covered a lot. I'm confident that we'll present the slides and take some time for questions, but I would really encourage you, and we'll share these slides again afterwards, dig into these articles, which are going to be able to see much more around all the plots and things that I showed. 3 of them are peer-reviewed publications. This last one is very close. We're just trying to get it over the finish line, and that should be out soon too. Thanks so much for your time, and we would really like to take some questions now and understand what you're all excited about.
Awesome. Thank you so much, Jack. If anyone has any questions, feel free to drop them into the Q&A. One of the first questions here is, why have pangenome references struggled with adoption if you all have been pushed them for the last decade?
Yeah, it's a great question. I think it comes down to a few things, and what I was hinting at in the POC. The fact that you can call more variants, or you can call things in difficult to call areas, is important. But since they're novel, you're not necessarily going to have an agreed upon impact or consensus around that variant. So is that a variant of unknown significance? Is that likely pathogenic? What does that mean? So we're kind of pulling out these results in unexplored areas and need people to show clinical impact to them. So we're working with a number of different groups along that way. And that's why we really want to have people try a POC to see, we believe this is really going to help them, but does it matter for the particular application that they have?
Awesome. Another question that came in, what disease areas may benefit particularly from pangenome references?
Yeah, that's a good question. I think one of the areas that may benefit is working specifically in pediatrics or other rare diseases where you don't necessarily have you won't have, fundamentally, you won't have as many patients as you would have for, say, non-small cell lung cancer. So there it really matters more and more that you have accurate, clean, high signal to noise ratio variants that you can depend on, because you're not going to be able to just pull them out of the noise by having 10,000 kids in your study.
Perfect. And then another question I see here, is there a potential trade-off in the necessary read depth that is required for pangenome aligners versus linear aligners?
Yeah, that's an interesting question. So I didn't have it in this presentation, but there have been some groups who started to look at, with their linear aligners, they were doing 90x sequencing on tumor samples. And they looked at what would happen. I think they did this artificially, actually, just subsampling it. But what if they only use 30x, or what if they only use 10x coverage depth? What they found is a pretty similar performance with graph at with pangenome based references at, say, 10x versus linear methods at 90x. I think there's a lot of work to be done there, but there could be a way to look at different trade-offs in the sequencing technology that you use once you could validate that, hey, you only actually need 30x, you don't need 90x to be able to move forward in this particular population in this particular indication.
Perfect. Alrighty. If any other questions remain, please do drop them in the Q&A portion. I'll just give about 30 seconds to see if we have any others trickle in. One quick reminder before we close out to mark your calendars for December 11th to learn about all things AI with Tatiana Kovacevic, who has been at some conferences, and so excited for that presentation coming up in December right before the holidays. Not seeing any other questions flowing in. Thank you so much to everyone for joining. Jack, thank you for your knowledge and insights here. And we wish you all a wonderful day and chat soon.